The last part we finished up with adaptive immunity to fungi, and now we are continuing with the immunity to virus. So we continue. Viruses are obligatory intracellular microorganism that uses component of nucleic acid and protein synthetic machinery of host to replicate. Viruses typically infect various cell types of receptor mediated endocytosis after binding to normal cells surface molecules. Viruses can cause tissue injury and disease by any of the several mechanisms. Viral replication interferes with normal cellular protein synthesis and function and lead to the injury and ultimately death of the infected cells. This results in one type of cytopathic effect of viruses, and the infection is said to be lytic because the infected cell is lysed. So viruses can stimulate inflammatory response and cause damage to tissues. Viruses may also cause latent infection, discussed later. So as you see, this is the just diagram we will see later on with a description of role of innate immunity and T helper cells defense against fungal. This was already done. So we continue. Innate and adaptive immune response to virus are aimed at blocking infection and eliminating infected cells. Innate immunity to viruses. The principal mechanism of innate immunity against viruses are inhibition of infection by type 1 interferons and natural killer cell mediated killing of infected cell. Infection by many viruses are associated with the production of type 1 interferons by infected cells and by plasmacytoids, dendritic cell, and macrophage responding to viral product. Several biochemical pathway triggers interferons productions. These include recognition of viral RNA and DNA by the endosomes toll-like receptor, and activation of cytoplasmic reg-like receptor and the STING pathway by viral RNA and DNA respectively. These pathways converge on the activation of protein kinases, which in turn activate IFR transcription factor that stimulates interferon genes transcription. The type 1 interferons inhibit viral replication in both infected and uninfected cells. The mechanism by which these cytokines block viral replication were discussed in chapter 4, figure 418. Type 1 interferon also stimulates the production of other host proteins that interfere with the viral replication and are called restriction factor. The role of restriction factors in defense against viruses is best understood for HIV. Natural killer cells kills viruses infected cell and are an important mechanism of immunity against viruses, especially DNA viruses, herpes viruses, human papilloma viruses, and others, early in the course of the infection before adaptive immune response have developed. Class 1 MHC expression is often shut off in virus-infected cells as an escape mechanism from cytotoxic T cells. This enables natural killer cell to kill the infected cell because the absence of class 1 releases natural killer cell from the normal state of inhibition. Viral infection may also stimulate expression of activated natural killer cells ligand on the infected cell. Adaptive immunity to viruses. Adaptive immunity against viral infection is mediated by antibody that blocks viruses binding and entry into the host cell, and by cytotoxic T lymphocytes, which eliminate the infection by killing the infected cell. The most effective antibodies are high affinity antibody produced in the T-dependent Gemino-center reactions. Antibodies are effective against viruses only during extracellular stages of lives of these microbes. Viruses will be extracellular when they first enter the body before they infect the host cell, or when they are released from the infected cell by virus budding, or if the infected cell die. Antiviral antibodies bind to viral envelope or capsid antigen and function mainly as neutralizing antibodies to prevent virus attachment and entry into host cells. Thus, antibodies prevent both initial infection and cell-to-cell spread. Secreted antibody, especially of IgA isotypes, are important in neutralizing viruses within the respiratory and intestinal tract. Oral immunization against polioviruses work by inducing mucosal immunity. In addition to neutralization, antibody may opsonize viral particles and promote their clearance by phagocytes. Complement activation may also participate in antibody-mediated viral immunity, mainly by promoting phagocytosis and possibly by direct lysis of viruses with the lipid envelope. So we have here the figure 16.8, this innate and adaptive immune response against viruses. A is kinetic innate and adaptive response to virus infection. So we have the type 1 interferons, viruses, they activated, they have neutralization, protection against the infection. Then B, mechanism by which innate and adaptive immune prevent and eradicate viral infection. So here we have the natural killer cells. If infected cell have to be killed, killing the infected cell. Sometimes cytotoxic T lymphocytes, CD8, also see the infected cell and kill. This can lead to the eradication. So the first one for antibodies for antiviral state, then neutralization in adaptive. Here is antiviral for innate and adaptive is neutralization. Then in innate is killing and also in the adaptive is killing. The importance of humoral immunity in defense against the viral infection is supported by the observation that resistance to particular virus induced by either infection or vaccination is often specific for serological antibody-defined type of virus. An example is influenza virus, in which exposure to one serological type does not confer resistance to other serotypes of viruses. Neutralizing antibodies block viral infection of cells and spread of viruses from cell to cell, but after viruses enter the cells and begin to replicate intracellularly, they are inaccessible to antibodies. Therefore, humoral immunity induced by previous infection or vaccination is unable to protect individual from viral infection, but cannot by itself eradicate established infection. Elimination of viruses that reside within cell is mediated by cytotoxic T lymphocytes, which kills the infected cells. As we have mentioned in the previous chapters, the principal physiological function of cytotoxic T lymphocyte is surveillance against viral infection. Most viruses, specifically cytotoxic T lymphocyte, are CD8 T cells that recognize cytosolic, usually endogenous synthesized viral peptide presented by MHC class I molecules. If the infected cell is tissue cells and not an antigen presenting cells, such as the dendritic cell, the infected cell or viral protein released from the cell may be phagocytosed by the dendritic cell, which process the viral antigen and present them to naive CD8 T cells to initiate the antiviral T response. We describe this process by cross presentation or cross priming in chapter 6. Full differentiation of CD8 T cell or cytotoxic T cells often require cytokine production produced by CD4 helper cells and or costimulatory expression by on antigen presenting cell. As discussed in chapter 9 and chapter 11, CD8 T cells undergo massive proliferation during viral infection, and most of the proliferating cells are specific for only few viral peptides. The activated T cells differentiate into effector cytotoxic T cells, which can kill any infected nucleated cell that is producing viral antigen in the cytosols and presenting peptide from these antigen on the surface class 1 MHC molecule. The antiviral effect of cytotoxic T cells are mainly due to killing of infected cell, but other mechanisms include activation of nucleases within infected cell that degrade viral genomes and secretion of cytokines such as interferon gamma, which activate the phagocytes. So this is the diagram showing figure showing the production of type of 1 interferon. So viral nucleotide acid is infected cells activate numerous pathways that lead to the production of antiviral type 1 interferons. If viruses enter the cell by the endosomes, the viral RNA and DNA can bind to toll-like receptor in the endosomal membranes, which induces signal that activate interferon response factor, interferon, and type 1 interferon gene expression during viral replication. Viral RNA in the cytosol can activate retinoic acid inducible genes like receptor, such as the melanoma differentiation associated gene 5, MDA5, and RIG1 and the viral double-stranded DNA can activate the SIG-STING pathway, thus leading to IFY or leading to the IRF activation and type 1 interferon gene expression, double-stranded cyclic guanosine monophosphate, adenosine monophosphate, and STING, which is stimulate of interferons. Many lines of experimental and clinical this was the diagram that we read, sorry. So we see the virus is coming in. We have in RNA, DNA, these are toll-like receptors that will present. So we have toll-like receptor 7, 8, 9, and 3. This can cause autophagy, nucleoside leakage. The RF7, RF1, RF3, they activate this. The transcription takes place, then to release the interferons and interferon alpha and interferon beta nuclei. So many lines of experimental and clinical evidence support the importance of cytotoxic T lymphocyte in defense against viral infection. Susceptibility to such infection is increased in patient and animal deficiency in T lymphocyte. Human and experimental animal can be protected against some virus infection by adoptive transfer of virus-specific restricted cytotoxic T cells. Viruses have developed numerous strategies to escape attack of CD8 cytotoxic T lymphocyte. These include blocking the processing and the presentation of antigen by the class 1 MHC pathway and shutting down CD8 T cells response by inducing a phenomenon of exhaustion. These evasion mechanisms are discussed later in the chapter. In a latent infection, viral DNA persists in host cell, but virus does not replicate or kill infected cells. Latence is a feature of infection by several viruses, especially DNA viruses, of Herpes viruses and pox virus family. In latent viral infection, the viral DNA may be integrated into DNA of infected cell, but no infectious virus is produced. The host immune response control but does not eliminate the infections. If the host immune system become defective for any reason, the viruses may be reactivated, causing significant clinical disease. Examples include shingle due to reactivation of latent varicella infection and cold sore due to reactivation of latent herpes simplex viruses. In some viral infection, tissue injury may be caused by a cytotoxic T lymphocyte. Some degree of immunopathology accompanying host T response to many, perhaps most of the virus infection. An experimental model of disease in which the pathological condition is primarily due to the host immune response is lymphocytic choriomeningitis virus. Infection in mice, which include inflammation of the spinal cord meninges and spinal cord meninges. Lymphocytic choriomeningitis virus infects meningeal cells, but it is non-cytopathic and does not injure the infected cell directly. The virus stimulates the development of virus-specific cytotoxic T cells that kill infected meningeal cells during the physiological attempt to eradicate the infection. Therefore, meningitis develop in normal mice with intact immune system, but T-cell deficient mice do not develop this disease and instead become carrier of the virus. This observation appears to contradict the usual situation in which immunodeficient individuals are more susceptible to infection disease than the normal individual. Hepatitis B viruses infection in human has the unusual features that immunodeficient persons who become infected often do not develop the disease but become carrier who can transmit the infection to other healthy persons. The liver of the patient with acute or chronic active hepatitis contain large number of CD8 T cells, and hepatitis viruses, specifically class 1 MHC restricted CTLs, and can be isolated from the liver biopsy specimen and propagated in vitro. These findings support the view that cytotoxic T lymphocyte response is the main cause of tissue injury in viral hepatitis. SARS-CoV-2 sometimes induces a systemic inflammatory reaction that contributes to the pathology and clinical manifestation of COVID-19. The inflammation is associated with the increased production of numerous cytokines. In some patients, the inflammation is severe and life-threatening. Severe disease is characterized by large myeloid cell-driven inflammation, with exuberant monocyte activation and cytokine production induced by viral RNA, possibly product of dead cells. Often there is severe lymphopenia with a decreased T cell number. The contribution of adaptive immune cells to inflammation in this disease is not well established, nor it is known why this particular coronavirus has a propensity to elicit such a reaction. It is also not clear why severe disease is seen in older individuals, especially in men with the pre-existing conditions such as type 2 diabetes. Immune response to viral infection may be involved in Immune response to viral infection may be involved in producing disease in other ways. Antiviral antibodies may promote activation of the macrophage and other myeloid cells through binding to Fc receptor and even enhance the entry of virus into host cell. This reaction, called antibody-mediated enhancement, has been postulated to increase lung inflammation associated with some viruses infections such as coronavirus, dengue, and respiratory syncytial virus. A consequence of persistent infection with some viruses such as hepatitis B is the formation of a circulating immune complex composed of viral antigen and specific antibodies. These complexes are deposited in the blood vessel and lead to the systemic vasculitis. Some coronavirus that causes severe life-threatening disease may induce endothelial damage and activate coagulation, thus inducing the local thrombus formation and tissue damage. Some viral protein contain amino acid sequences that are also present in some self-antigens. It has been postulated that because of this molecular mimicry, antiviral immunity can lead to the immune response against self-antigen. Immune evasion by viruses. Viruses have evolved numerous mechanisms for evading host immunity. Viruses can alter their antigen and thus no longer a target of immune response. The antigen affected are the most commonly surface glycoprotein that are recognized by antibody, but T cell epitope may also undergo variation. The principal mechanism of antigenic variation are point mutation and the reassessment of RNA genomes in RNA viruses, leading to antigenic drift, antigenic shift. These processes are of great importance in the spread of influenza viruses. There are two antigens of the virus that are the two major antigens of viruses are trimeric viral hemagglutinin, the viral spike protein, and neuroaminidase. Viral genomes undergo mutation in genes that encode this surface protein, and the variation that occurs as a result is called antigenic drift. Alternatively, the segment of RNA genomes of virus strain influenza viruses that normally inhibit different host species can recombine in host cell, and these reassortment virus can differ quite dramatically from prevalent strain. Reassortment of viral genes result in major changes in antigenic structure called antigenic shift, which create a distinct virus that as avian flu or swine flu viruses. Because of antigenic variation, a virus may become resistant to immunity generated in the population by previous infections. The influenza pandemic that occurred in 1957 and 1968 were due to different strains of viruses, and H1N1 pandemic in 2009 was due to a strain in which the strand of RNA genome was a reassessment among strains endemic in pigs, fowls, and humans. Subtler viral variants arise more frequently. There are so many serotypes of rhinoviruses that vaccination against the common cold may not be feasible preventive strategy. HIV-1, which causes AIDS, is also capable of tremendous antigenic variation because of high error rate in reverse transcriptions of its RNA genome during viral replications. In this situation, prophylactic vaccination may be directed against invariant viral proteins. So we have table 16.3, mechanism of immune evasion by viruses. Mechanism of immune evasion, example. So there is antigenic variation, is more seen in influenza, HIV, and rhinoviruses. Inhibition of antigen processing, blockage of TAP transporter, removal of class 1 molecules from endoplasmic reticulum, CMV, HESV, and also remember HIV may be involved. Production of decoy MHC molecule to inhibit natural killer cell. This is cytomegalovirus in murines. Production of cytokine receptor homologue. This is Yersinia, poxviruses, and cytomegalovirus. Production of immunosuppressive cytokines, e.g., for Epstein-Barr virus, which is separate a lot of interleukin-10s. Infection and death or functional impairment of immune cells, mostly in HIV. Inhibition of complement activation, recruitment of factor H, incorporation of CD59 in viral envelopes. This is in HIV, Yersinia, human cytomegalovirus as well. Inhibition of innate immunity, inhibition of access to RIG, RNA sensor inhibition of PKR signaled by interferon cell. This is mostly in Yersinia, HCV, and polio. So those are the ones that can cause that particular problem. Some viruses inhibit the ability of the host to induce the antiviral state. The coronavirus SARS-CoV, MERS-CoV, and SARS-CoV-2 can all shut off the host type 2 interferon's response. Among the protein produced by SARS-CoV-2, 10 and of 29 are dedicated to dampening or prevent host type 1 interferon production. They do so by various means. Some viral protein modify viral RNA to make it more closely resemble host messenger RNA by adding a version of 7-methylguanosine cap and by adding the 2 prime methyl group to the ribose in the next nucleosides. This prevents recognition of RIG1 and MDA5. Other proteins attenuate the transcription of host type 1 interferons. The importance of type 1 interferons as an antiviral mechanism is further highlighted by the observation that individual who makes antibody against their own type 1 interferons or have genetic defect in interferon production or signaling are increased risk of developing severe COVID-19 following the infection with SARS-CoV-2. Some viruses inhibit class 1 MHC associated presentation in cytotoxic protein antigen. Viruses make a variety of protein that block different steps in antigen processing, transport, and presentation. Inhibition of antigen presentation block the assembly and the expression of stable class 1 MHC molecule and display a viral peptide. As a result, cells infected by such a virus cannot be recognized or killed by CD8 T cells. As discussed earlier, natural killer cells are activated by infected cells, especially in the absence of class 1 MHC molecule. Some viruses may produce protein and act as ligand for natural killer cells inhibitory receptor and thus inhibit natural killer cell activation. So this is influenza virus that you see how it is changing generation of new influenzas. So some viruses again produces molecular that inhibit the effector phase of immune responses. Poxvirus encode a molecular that secreted by infected cells and bind to several cytokines, including interferon gamma, tumor necrosis factor, interleukin-1, interleukin-18, and chemokines. The secreted cytokines binding protein may function as competitive antagonists of the cytokines. Epstein-Barr viruses produce a protein that is homologous to the cytokine interleukin-10, which inhibit activation of macrophage and dendritic cells and may thus suppress cell-mediated immunity. These examples probably represent a small fraction of immunosuppressive viral molecules. Identification of these molecules raises the intruding possibility that viruses can have acquired gene encoding endogenous inhibition of immune response during their passage through human host and have thus evolved to infect and colonize human. Some chronic viral infections are associated with failure of cytotoxic T cell lymphocyte response, called exhaustion, which allow persistence. Studies of chronic infection with the LCMV in mice have shown that this type of immune deficient may result from persistent antigen stimulation leading to upregulation of T cell inhibitory receptors such as PD-1, programmed cell death protein 1. There is evidence for CD8 T cells exhaustion in chronic human viral infection, including HIV and hepatitis virus infection. The physiological importance of PD-1 mediated T cells inhibitory may be that it limits the pathology caused by a strong immune response to viruses. Viruses may infect and either kills or inactivate immunocompetent cells. The obvious example is HIV, which survived by interfering and letting CD4 T cells. They induce the immune response to protein antigen. Immune to parasite. Parasites include single-celled protozoa, complex multicellular worms, helminth, and ectoparasite, e.g. ticks and mites. Parasitic infection are major health problems, particularly in low-income countries. It is estimated that approximately 30% of the world population suffer from the parasitic infection. There are about approximately 200 million new malaria cases each year worldwide, approximately 400,000 deaths annually. The magnitude of this public health problem is the principal reason for the great interest in immunity to parasite and of the development of parasitology as distinct branches of immunology. Most parasites go through complex life cycle, part of which occur in human or other vertebrate, and part of which occur in intermediate hosts such as flies, ticks, and snail. Humans usually are infected by bites from the infected intermediate host or by sharing particular habit with the intermediate host. For instance, malaria and trypanosomes are transmitted by insected bites. Schistosomiasis is transmitted through exposure to water, in which infected infected snails reside. Many parasitic infections are chronic because of weak innate immunity and ability of the parasite to evade or resist elimination of adaptive immune response. Furthermore, many antiparasitic drugs are not effective at killing the organism. Individuals living in the endemic areas require a repeated chemotherapy because of continued exposure. And such treatment is often not possible because it is expensive and logistic problems. Innate immunity to parasite. Although the different protozoa and helminth parasite have been shown to activate different mechanisms of innate immunity, these organisms are shown to be able to survive and replicate in their host because they are well adapted to residing host defense. The principal innate immune response to protozoa is phagocytosis, but many of these parasites are resistant to phagocytes killing and may even replicate within macrophage. Some protozoa expresses surface molecules that are recognized by toll-like receptor and activate phagocytes. Plus, Plasmodium species, the protozoa that responds for malaria, Toxoplasma gondii, the agent that causes toxoplasmosis, and the Cryptosporidium species that causes the diarrhea disease in HIV infected patient, all expresses glycoprotein that can activate toll-like receptor 2 and toll-like receptor 4. Eosinophilia contribute to the innate response to helminths by releasing granule contents that are capable of destroying worm integuments. Phagocytes may also attack helminth parasite and severe and secrete microbial substance to kill organism. However, many helminths have thick integument that make them resistant to the cytocidal mechanism of neutrophil and macrophage, and they are too large to be ingested by these phagocytes. Some protozoa and helminths activate an alternative pathway of complement but they have also developed effective strategies to evading complement system. So these are the structure. This is the inhibition of proteasomal activities. So cytosolic protein Then these are released in the tap, they have to go. I think this is the mechanism for virus, not for parasite. This is the mechanism of which viruses inhibit recognition by CD8 T cells and natural killer cell. The pathway of class 1 major histocompatibility complex associated with antigen presenting is shown with the example of viruses in it. So immune response to disease causing parasite. So we are speaking of, when you're speaking of protozoa, for example, malaria species causes disease malaria. Principal mechanism protective is antibody and CD8 cytotoxic T lymphocyte. Leishmania dovans, it causes leishmaniasis, mucocutaneous or disseminated. Then CD4 T helper cell activate macrophage to kill phagocytosed the parasite. Trypanosoma Crohn's African trypanosomosis is mostly principal mechanism is antibody. Entamoeba histolytica, the causes disease amoebiasis. It's mostly depend on antibody and phagocytosis. Metazoa, Schistosoma species, schistosomiasis, killing by eosinophil macrophage. Filaria, e.g., Wuchereria bancroft, is cell-mediated immune role of antibodies. Adaptive immune to parasite. Different protozoa and helminths vary greatly in their structural and biochemical properties. Life cycle and pathogenic mechanism. Different protozoans and helminths vary greatly in their structural and biochemical process, life cycle, and pathological mechanism. It is therefore not surprising that different parasites elicit distinct adaptive immune response. Some pathogenic protozoa have evolved to survive within host cell. So a protective immunity against these organisms is mediated by mechanisms similar to those that eliminate intracellular bacteria and viruses. In contrast, the metazoas such as helminths survive in the extracellular tissue, and their elimination often depend on special types of antibody response. The principal defense mechanism against protozoa that survive within macrophage is cell-mediated immunity, particularly the macrophage activated by T helper 1 cell-derived cytokines. Infection of mice with the L major, a protozoa that if survive within endosomes of macrophage, it assesses how dominance of Th1 and Th2 response determine the disease resistance or susceptibility. Recent resistance to the infection is associated with activation of Leishmania species specific Th1 cells, which produce interferon gamma and thereby activate macrophage and to destroy intercellular parasite. Conversely, activation of T helper 2 cells by protozoa results in increased parasitic survival and exacerbation of lesion because T helper 2 cytokines inhibit classical macrophage activation. Most inbred strains of the mice are resistant to infection with L major, but bred bulb some related to the strain of the mice are highly susceptible and die if they are infected with high dose of parasites. The resistant strain produce large amount of interferon gamma in response to Leishmania's antigen, whereas the strain that are susceptible to fatal Leishmaniasis produce an interleukin-4 in response to their parasite. Promoting the T helper 1 response or inhibiting the T helper 2 response in susceptible strain increases the infection. The mechanism of this striking disease between strain and mice is not defined. find. So this is another diagram that we see role of T cell and cytokines in determining the outcome of infection. Naive CD4 T cell may differentiate into T helper 1 cells which invade the phagocytes to kill ingested microbes, and T helper 2 cells which inhibit this classical pathway of macrophage activation. The balance between these two T cell subsets may influence the outcome of infection, as illustrated in Leishmania infection in mice. Host mouth strain developed T helper 1 response against the parasite and effectively clear the organism, but bulb mice developed strong Th2 response and succumbed to infection. So Leishmania major infection in strain. So naive CD4 T cell, it is when it meets the Leishmania, it causes the activation of T helper 2 to T helper 1. Then it produces a lot of interferon. Interferon leads the classical macrophage activation mediated immunity. Again, when you're speaking of Leishmania major infection in the bulb mice naive CD4, then this causes the production of T helper 2, and this T helper 2 releases interleukin 10, interleukin 4, 13. Then it inhibits classical macrophage activation. The role of T cells and cytokines in determining the outcome of infection. Naive CD4 T cells lymphocyte may differentiate into helper 1 cells which activate the phagocyte to kill ingested microbes, and T helper 2 cells which inhibit this classical pathway of macrophage activation. The balance between these two T subsets may influence the outcome of infection, and as illustrated by Leishmania infection in mice, most mouse strain develop Th1 response against the parasite and effectively clear the organism, but bulb mice develop strong T helper 2 response and succumb to the infection. Protozoa that replicate inside various host cell and lyse these cells stimulate specific antibody and cytotoxic T lymphocyte response. Similar to cytopathic viruses, an example of organism is malaria parasite, mainly reside in the red blood cells and in the hepatocyte during its life cycle. It was thought for many years that antibodies were major protective mechanism against malaria, and early attempt at vaccinating against this infection focused on generative process protective antibodies. It is now apparent that cytotoxic T cells response against parasite reside in the hepatocyte is an important defense mechanism against the spread of intracellular protozoa. So cytokine interferon gamma has been shown to be protective in many protozoan infections, including malaria, toxoplasmosis, and cryptosporidosis. Defense against many helminth infection is mediated by activation of T helper 2 cell, which results in the production of IgE antibodies and activation of eosinophil. Then helminth stimulates differentiation of naive CD4 T cell to T helper 2 subset of effector cells, which secrete interleukin 4, interleukin 5, and interleukin 4 stimulate production of IgE, which bind to the FCE receptor of the mast cell, and interleukin 5 activate eosinophils. The combined action of the action of the mast cell and eosinophilia also contribute to the expression of the parasite from the intestine, although exactly how exactly how they contribute remain poorly defined. The expression of some intestinal nematode may be due to T helper 2 dependent mechanisms T that do not require IgE, such as increased mucus production and parasites caused by T helper 2 cytokines. Adaptive immune response to parasite can also contribute to the tissue injury. Some parasites and their products induce granulomatous response with concomitant fibrosis. Schistosomiasis monsoon eggs deposited in the liver stimulate CD4 plus T cells, which in turn activate macrophage and induce um, delayed type hypersensitivity reaction. Latent type hypersensitivity reaction result in these granulomas, especially in mice, is associated with T helper 2 cells. Granulomas are generally induced by T helper 1 response against persistent antigen. Such T helper 2 induced granulomas serve to contain the cytosol eggs, but severe fibrosis associated with this chronic cell-mediated immune response lead to the cirrhosis, disruption of several venous blood flow in the liver, and portal hypertension. In lymphatic filariasis, lodging of the parasite to lymphatic vessel lead to the chronic cell-mediated immune reaction and ultimately to fibrosis. This results into lymphatic obstruction and the severe lymphoedema. Chronic and persistent parasitic infection are often associated with the formation of complex of parasite antigen and specific antibodies. The complex can be deposited in blood vessels and kidney, cause glomerulus, and produce vesicular nephritis, respectively, which is immune complex disease in complication. Immune complex disease is complication of schistosomiasis and malaria. Immune evasion by parasite. Parasite evade protective immunity by reducing their immunogenicity by inhibiting host immune response. Different parasites have developed remarkably effective ways of resisting immunity. So, mechanism of immune evasion by parasite. We have antigenic variation, Trypanosoma and Plasmodium, acquire resistance to complement cytotoxic and T lymphocyte. We have Schistosoma, inhibition of host immune response. This is Furario, second to lymphatic obstruction, Trypanosomes. Then antigen shedding, which is an Entamoeba. Parasites change their surface antigen during their life cycle into vertebral host. Two forms of antigenic variation were defined. The first is a stage-specific change in antigen expression such that mature tissue stages of parasite produce antigen different from those of effective stage. For example, the infective sporozoite stage of malaria parasite is antigenically distinct from merozoite that reside in the host and are responsible for chronic infection. By the time the immune system has responded to infections, the sporozoites, the parasite have differentiated, expressing new antigen, and is no longer a target for immune elimination. A more remarkable example of antigenic variation in parasite is continuous variation of major surface antigens seen in African trypanosomiasis, such as Trypanosoma brucei, Trypanosoma rhodensis. Continuous antigen variation in trypanosoma is mainly due to the changes in expression of the gene encoding the major surface antigen. Infected patient shows waves of bloody parasitemia, and each wave consists of parasites expressing the surface antigen that is that is different from the preceding waves. Thus, by the time the hosts produce antibodies against the parasite, the antigenically different organ has been shown, antigenically different organ has been seen. One consequence of antigenic variation in parasite is that it is difficult to effectively vaccinate individual against this infection. Parasite because become resistant to immune effect of immune effector mechanism during their residence in vertebral host. Perhaps the perhaps Perhaps the best example are cytosome larvae, which travel to the lungs of infected animal, and during this migration develop a tegument that resists to the damage of complement and by CTL. Protozoa parasite may conceal themselves from the immune system either by giving inside host cells by developing cysts that are resistant to immune effectors. Some helminth parasites reside in intestinal lumens and are sheltered from the cell-mediated immune effector mechanism. Parasite may also shed their antigenic host, either spontaneous and after binding specific antibodies. The shedding of antigen render um, the parasite resistant to subsequent antibody-mediated attack. Amoeba histolytica is the protozoan parasite that shed antigen and can also convert to cystic form in lumen of large intestine. Parasite inhibit host response, immune response by by multiple mechanisms. T-cell energy due to parasite antigen has been observed in severe cystosomiasis involved in liver and spleen or in the viral infection. The mechanism by which the mechanism of immunological and responsiveness is in this infection are not well understood. In lymphatic filariasis, infection of lymph nodes, which with subsequent and architectural disruption may contribute to the deficiency immunity. Some parasites, such as Leishmania, stimulate development of regulatory T cells, which suppress the immune response enough to allow persistence of parasites. More non-specific and general immunosuppression is observed in malaria and African trypanosomiasis. This immune deficiency has been attributed to the production of immunosuppressive cytokine by activated macrophage and T cells and defect in T cell activation. The consequence of parasitic infections for healthy and economic development are devastating. Attempts to develop effective vaccine against infection have been actively pursued for many years. Although the progress have been slow, elucidation of the fundamental mechanism of immune response to and immune evasion parasite hold the promise for the future. Now let us look to the strategy of vaccine development. The birth of immunology as science dates back to Edward Jenner's successful in vaccination against smallpox in 1796. The importance of the prophylactic immunization against infectious disease is the best illustrated by the fact that worldwide programs of vaccination has led to the complete or nearly complete eradication of many of these diseases in many countries. The fundamental principle of vaccination is to administer a killed or attenuated form of infectious agent or component of microbe that does not cause disease but elicit an immune response that provides protection against infection by live or pathogenic microbes. The success of vaccination is in eradicating infectious disease depend on several properties of microbes. Vaccines are the most effective if the infectious agent does not establish latence, does not undergo antigenic variation, and does not interfere with the host immune response. It is difficult to effectively vaccinate against microbes such as HIV, which establish latent infection, is highly variable and inhibit host immunity. Vaccines are also the most effective against infections that are limited to human hosts and do not have animal reservoir. Most vaccines in use today work by inducing humoral immunity. Antibodies are the only immune mechanism that prevent infection by neutralizing and cleaning the microbes before they gain their foothold in the host. The best vaccines are those that stimulate the development of long-lived plasma cells that produce high affinity antibodies and memory B cells. These aspects of humoral immune response are best induced in geminocentric reaction, which requires help to provide which requires help to provide help which requires help provided by protein antigen specific CD4 T follicular helper cells. There are major challenges in developing effective vaccine against several important infections. The immunological correlate of protection are often poorly defined. Fundamental question on how the maximum stimulate the fundamental questions about how to maximally stimulate durable memory, effector T follicular cell, and long-lived plasma cell remain unresolved. Clinical expertise has taught us that longevity of vaccine-induced protection vary greatly, being lifelong with hepatitis B antigen vaccine and quite short with many others. The reason for this critical difference are known. It is hoped that continuing advances in basic immunology and method for analyzing immune response in human will lead to the answer to this question that will put vaccine development on a strong foundation of mechanism and the understanding. In the following section, we will illustrate the approaches to vaccination that have been tried and their major value and limitation. Attenuated and inactivated bacterial and viral vaccine. Some of the earliest first generation and most effective vaccines are composed of the intact microbes that are treated in such a way that they are attenuated or killed so they can no longer cause diseases while retaining their immunogenicity. The great advantage of attenuated microbial vaccine is that they elicit many of the innate and adaptive immune response, both humoral and cell-mediated, that pathogenic microbes would, and they are therefore pathogenic microbe would, and they are therefore the ideal way for inducing protective immunity. Live attenuated bacteria were the first shown by Louis Pasteur to confer specific immunity. The attenuated or killed bacteria vaccine currently in use generally induce limited protection and are effectively for only short period. Live attenuated vaccines live attenuated viral vaccines are usually more effective. Are usually effective for only short period. Live attenuated viral vaccine are usually more effective. Polio, measles, and yellow fever are three good examples. Earliest approach for producing such attenuated vaccine was as repeated passage in cell culture. More recently, temperature sensitive and gene deletion mutant have been generated to create attenuated viruses. Viral vaccine often induce long-standing specific immunity, so immunization of the children with a sufficient so immunization of children is sufficient for lifelong protection. The major concern with attenuated viral or bacterial vaccine is safety. The live attenuated oral polio vaccine has nearly eradicated the disease, but in rare cases, the virus in the vaccine is reactivated and itself causes paralytic polio. In fact, the success of worldwide vaccination is creating the problem that vaccine-induced disease, although rare, could become more frequently than natural occurring disease. This potential problem may be tackled by reverting to the killed virus vaccine to complete the eradication program. So we have vaccine approach, type of vaccine example. We have live attenuated or killed bacteria. We have Bacillus calmette-guerin (BCG). Live attenuated or killed virus. We have polio, influenza, and rabies. Subunit antigens: tetanus toxoid, diphtheria. Conjugate vaccine: Haemophilus influenza, pneumonia, pneumococcus. Synthetic vaccine: hepatitis, recombinant protein. Viral vector: clinical trial of human SARS-CoV-2 spike protein made by human and chimpanzee adenovirus. Then is DNA vaccine, clinical trial ongoing on several infections. Messenger RNA vaccine approved for COVID-19. A widely used inactivated vaccine for considerable public health importance is influenza vaccine. Influenza vaccine grown in the chicken eggs are used in two types of vaccine. The most common vaccine is a trivalent inactivated killed vaccine that are used for short flu that is given intramuscularly. Three of the most frequently encountered influenza strains are selected every year and incorporated in this vaccine. A second type of influenza vaccine involves the same three strains, but the vaccine is made up of live attenuated viruses and is used as nasal spray. Two of the major limitations of current influenza vaccine is that they do not induce broadly neutralizing antibody that recognize multiple strains of the virus and antibody-mediated protection is short-lived. Purified antigen subunit vaccine. These second-generation vaccines were produced to eliminate safety concerns associated with the attenuated microbes. Subvaccines are composed of antigen purified from the microbes and inactivated toxin, and are usually administered with an adjuvant. One effective use of purified antigen vaccine is for the prevention of disease caused by bacteria toxin. Toxin can be rendered harmless without loss of immunogenicity, and such toxoid induce strong antibody response. Diphtheria and tetanus are shown are two infections whose life-threatening. Diphtheria and tetanus are two infections whose life-threatening consequences have been largely controlled because of immunization of children with the toxoid preparations. Vaccine composed of bacteria polysaccharide antigen are used against pneumococcal and influenza and the Haemophilus influenza. Because of polysaccharide are T-dependent antigen, they can tend to elicit low affinity, low antibody response, and a poor immunologic in infant. Poor immunologic in infant mount strong T-cell independent antibody response. High affinity antibody response may be generated against polysaccharide antigens even in infant by coupling the polysaccharide protein to form conjugate vaccine. This vaccine at least help us T cells to simulate a germinal-center reaction, which would not occur with simple polysaccharide vaccine, such a vaccine would like Hapten carrier conjugate and practical application of the principle of T and B cell cooperation. The current used H influenza, pneumococcal, and meningococcal vaccine are conjugate vaccine. Purified protein vaccine stimulate helper T cells antibody response, but they do not generate potent cytotoxic T cells. The reason for poor cytotoxic development is that exogenous proteins and peptides usually enter the class 2 MHC pathway of antigen presentation, except in the special situation of cross presentation. As a result, protein vaccines are not recognized efficiently by class 1 MHC, class 1 CD8 T cells. Synthetic antigen vaccine. A goal of vaccine research has been to identify most immunogenic microbial antigen or epitope. To synthesize these laboratory and use the synthetic antigen as vaccine. It is possible to deduce the protein sequence of microbes and addition antigen from nucleotide sequence data and to prepare large quantities of proteins by recombinant DNA technology. Vaccine made from DNA-derived antigen are now in use for hepatitis B viruses, hepatitis V. In case of most widely used HPV vaccine, which was developed to prevent cancer caused by virus recombinant in viral proteins. The HPV, interleukin HPV, there are 6 strains, 6, 11, and 6 and 18 are made in the yeast and combined with the adjuvant. So they are common causes of warts and HPV in 16 to 18 years old ladies. Strain among often linked to cervical cancer. We have live viral vaccine involving recombinant. So here another approach of vaccine development is introduce gene encoding microbial antigen into a non-cytopathic virus and to infect individual with this virus. Thus, viruses serve as source of the antigen in an enucleating individual. The great advantages of viral vector is that they like other they, like other live viruses, induce full complement of immune response, including strong cytotoxic T lymphocyte response. This technique has been used in most commonly in vaccinia viruses vector and more recently with the Canarypox viral vector, which are not pathogenic in human. Inoculation of such a recombinant viruses into any species of animal induce both humoral and cell-mediated immunity against the antigen produced by the foreign genes, e.g., foreign genes, and the cause against antigen of viral vector as well. A potential problem with recombinant virus is that the viruses may infect host cell, and even though they are not pathogenic, they may produce antigen that stimulates cytotoxic T cell response that kills the infected host cell. Also, the non-pathogenic viruses could recombine with host viruses or gene sequence and become virulent. These safety concerns have limited widespread use of viral vectors for vaccine delivery. One approach that overcome many of these issues are concern. One of the approach that overcome many of these issues and concern is that the use of live recombinant hybrid vaccine that are non-replicating. An adenovirus 26 vector human greatly lack antibodies to these adenoviruses, and the chimpanzee adenovirus vector has been used to generate vaccines to a number of viruses including Ebola viruses, Zika viruses, and SARS-CoV-2. Non-replicating adenovirus infect numerous host cells and thus produce a significant amount of viral antigen. DNA is an interesting method of vaccination was developed on the basis of unexpected observation. Inoculation of a plasmid containing complementary DNA encoding the protein antigen lead to humoral and cell-mediated immune response as to antigen. It's likely that antigen-presenting cells such as the dendritic cell are transferred by plasmid, and the copy of DNA is transcribed and translated into the immunological protein that elicits specific responses. Bacteria plasmid are rich in unmethylated CpG nucleotides that are recognized by T receptor TLR9 in dendritic cells and other cells, thereby eliciting an innate immune response that enhances adaptive immunity. Therefore, plasmid DNA vaccine will be effective even when administered without adjuvant. The ability to store the DNA without refrigeration for the use in the field also make this technique promising. However, DNA vaccines have not been as effective as hoped in clinical trial, mainly because the first generation of this vaccine did not provide the adequate amount of human immunogen. Studies with fewer vectors for DNA vaccination are currently in progress. Messenger RNA vaccine. So another relatively recent mode of vaccination uses messenger RNA encoding microbial antigen. The main advantage of messenger RNA vaccine are too easier with which they can be rapidly developed. The ability to bypass the need of large-scale manufacture or purification of protein antigen, thereby greatly reduced cost, and the ability to combine messenger RNA encoding many different protein antigen from pathogen into single vaccine. Adjuvant and into single vaccine. Although initial attempt to use messenger RNA was unsuccessful, largely because of a stability issue. Number of recent advances have made messenger RNA vaccination a practical modality. One major advance in modification of messenger RNA itself. This modification includes addition of synthetic 5' CAR and a long poly-A-tailed increase stability, altering 5' and 3' untranslated region of the messenger RNA to enhance the translations and stability, and codon opsonization of coding portion to enhance transmissibility. Current messenger RNA vaccine for COVID-19 retains some ability to activate innate immunity by triggering RNA sensor. The messenger RNA is encapsulated in lipid nanoparticles that facilitate uptake by cells, including dendritic cells, and also function as an adjuvant. Another approach for messenger RNA vaccine are not yet in clinical use involves linking the messenger RNA to a modified alpha virus RNA genome that allows for self-replication, thus allowing many copies of the vaccine to be generated in the recipient cell. Adjuvants and immunomodulators. The initiation of T-cell dependent immune response against proteins antigen require that the antigen be administered with adjuvant. Most adjuvant elicit innate immune response with increased expression of costimulators and production of cytokines such as interleukin 12 that stimulate T-cell growth and proliferation. Heat-killed bacteria are powerful adjuvant that are commonly used in experimental animals. However, severe local inflammation that such bacteria trigger the prerequisite use of an adjuvant is inhuman. Much effort is currently being devoted to develop a safe and effective adjuvant for clinical use. Only few are approved for patients. Aluminium hydroxide gel, which appears to promote mostly B cell response. A bacterial product, monophosphoryl A, alone or with aluminium, and many other molecules. Passive immunization. Protective immunity also can be conferred by passive immunization, for instance, by transfer of specific antibody to clinical situation. Passive immunization is most commonly used to rapidly treat the potential fatal disease caused by toxins such as tetanus and the protection from rabies, hepatitis, and SARS-CoV-2. Antibody against snake venom can be life-saving when administered after the poisonous snake bite. Covalescence plasma have been used in case of Ebola and COVID-19. Recombinant monoclonal neutralizing antibodies are now utilized as therapy for COVID-19. Passive immunity using current approach is short-lived because of the host does not respond to immunization, and protection lasts only as long as injection antibody persists. Moreover, passive immunization does not induce memory, so an immunized individual is not protected against subsequent exposure to toxin microbes. However, based on successful identification of human broadly neutralizing monoclonal antibodies against pathogens such as HIV and flu vaccine, newer attempts of longer to long-term passive immunization using the process called vectored immunoprophylaxis have been also developed. In this approach, adeno-associated viral vectors are used to introduce cloned human Ig heavy and light chain genes for their neutralizing antibody into human subject. The goal is to have injected human synthesize a specific protective broadly neutralizing antibody for an extended period. Clinical trial have been initiated. So, summary, we have been studying this topic, and the summary is the interaction of immune system with infectious organism is dynamic interplay of the host mechanism aimed at eliminating infectious and microbial strategies designed to permit survival in the face of powerful defense. Different types of infectious agents simulate distinct type of immune response and have evolved unique mechanism for evading immunity. In some infections, the immune response is the cause of tissue injury and disease. Innate immunity against extracellular bacteria is mediated by phagocyte and the complementary system alternative or pathway. Innate, the principal adaptive immune response against extracellular bacteria consists of specific antibodies that opsonize the bacteria for phagocytosis and activate the complement pathway. The toxin produced by such bacteria are neutralized by specific antibodies. Some bacteria toxins are powerful inducers of cytokine production, and cytokine account for much of the systemic disease associated with severe disseminated intravenous infection with these microbes. Innate immunity against intracellular bacteria is mediated mainly by macrophage. However, intracellular bacteria are capable of surviving and replicate within host cells, including phagocytes, and because they have developed mechanism for resisting degradation within phagocytes. Adaptive immunity against intracellular bacteria is principally cell-mediated and consists of activation of macrophages by CD4 T cells as well as killing infected cells by CD8 cytotoxic T lymphocytes. The characteristics of phagocytic response to infection by intracellular bacteria is granulomatous inflammation. Protective response to fungi consists of innate immunity mediated by neutrophil and macrophage adaptive cells mediated immunity mainly involving T helper 17 cells and humoral immunity. Fungi are usually readily eliminated by phagocyte because of which disseminated fungal infection are seen mostly in immunodeficient persons. Innate immunity against viruses mediated by type 1 interferons and natural killer cell. Neutralizing antibody protect against viruses entry into the early in the course of the infection, and later if the viruses are released from killed infected cells. The major defense mechanism against established infection is cytotoxic T lymphocyte mediated killing of infected cells. Cytotoxic T lymphocyte may contribute to the tissue injury even when infectious viruses is not harmful by itself. Viruses evade immune responses involving the antigenic variation, blocking type 1 interferon gamma production or action, inhibiting the inhibition of antigen presentation, inactivation of T cells, and production of immunosuppressive molecules. Parasites such as protozoa and helminths give rise to chronic and persistent infection because innate immunity against them is weak, and parasites have evolved multiple mechanisms for evading and resisting specific immunity. The structural and antigenic diversity of pathogenic parasites is reflected in the heterogeneity of the adaptive immune responses that they elicit. Protozoa that live within host cell are destroyed by cell-mediated immune, whereas the helminths are eliminated by eosinophil-mediated killing. Parasite this parasite evade the immune system by varying the antigen during the residence in vertebrate host, by acquiring resistance to immune effectors mechanism, and by masking and shedding the surface region. Vaccination is a powerful strategy for preventing infections. The most effective vaccines are those that stimulate the production of high high affinity antibodies and memory cells. Many approaches for vaccination are in clinical use and being carried out by virus infection, being tried for virus infection. This is the last part of this topic. Yeah, and up there.